Thank you all for coming on board and kind of presenting your work here. Um, we're going to move next to,
Thank you very much, Javier, and thank you to the organizers, to Pradeep, and for asking me to present. And it's great to see all the talks so far. It's been really interesting. And I actually did my PhD in biostat at Pittsburgh.
Hey.
With with my advisors were um, Rob Crafty and Stuart Anderson, and I worked with Howard Eisenstein and Dana Tudoroscu. So it's great to see their names coming up, and it's great to be back in Pittsburgh again in a virtual sense and see what's going on there. Let me do presenter mode. Let's see. Okay, so yeah, I'll discuss, data harmonization for MRI, and it's great to see there's a lot of interest in this topic right now. Yeah, I'm a postdoc at Pensive, by the way, which does a lot of work in this area. As we all probably know, that we're seeing a lot more multi-site and multi-scanner neuroimaging studies. And just to define some terms, we have what are called batch effects, or maybe site effects or scanner effects, which represent unwanted technical as opposed to biological variability related to the site and scanner where the data was acquired. And this distinction kind of maybe comes from the genomics literature originally between technical and biological variability. So I think it's relevant here as well. And data harmonization, I don't know if it's precisely defined anywhere, but I'm just a general definition. It's, removing or accounting for this unwanted technical variability. Right, so, and we're also seeing more complex, longitudinal designs as well, where a scanner might be changing, within a subject. So, and here's just an example of many of the multi-site studies that are out there now. Just a selection. So aggregating data across sites and scanners can give greater statistical power, but we need to be able to deal with the site and scanner effects. So for example, here's an example from the Baltimore longitudinal study of aging, where we have the same subject, consecutive scans. I'm not really sure the timing precisely between these, but there are different field strengths, different manufacturers, different pulse sequences. And you can see the difference in the contrast between the different tissue types in these images. And what can happen in downstream analyses is, like, if we're extracting, let's say, cortical thicknesses from images, this is an example from ADNI data, we see additive effects, so shifts in the mean across the scanners, after adjusting for known, biological variability. Here in this picture, these are colored by the field strength. So it's known that, higher field strength yields sort of greater cortical thicknesses. And then at the bottom, there's a picture of, where in the brain we saw these most prevalent additive effects in this ADNI data set. And then we can also talk about multiplicative site and scanner effects. So the variability might differ in the residuals sort of across scanners. And so here is an example where we saw there was an association with the vendor or the manufacturer of the scanner, with some being more variable. And you can see where in the brain that was most prevalent in the yellow areas. So there's like a lot of different potential causes of site and scanner effects, and we want to minimize scanner effects, so we want to address all of these wherever we can. So first of all, the study sample characteristics should ideally be balanced across site and scanner. If we want to separate out, we don't want confounding of, we want to be able to measure the scanner effect and not have it be attributable to, like, differences in age or diagnosis across the scanner. You know, different acquisition protocols can yield different scanner effects. Scanner hardware, magnetic field inhomogeneities can differ across scanners, and just even the magnetic field characteristics of the scanner location. Like I've heard an example of, there was actually a potted plant or something outside of the scanner room where the scanner was located, and that was distorting the magnetic field, sort of, which was something they found out later. And you know, maybe there's sort of weather effects, I don't know, electrical storms and stuff. And then we have a scanner software, image processing pipelines that can differ, and you know, within scanner there could be scanner drift, and so on. And so some of these things we can measure, and other things are very, you know, harder to keep track of. And there's a lot of work sort of that's been kind of dissecting, you know, the sources of all the scanner effects. There's some example references. So in addressing these, as I mentioned, we can do that at various points along the data acquisition and processing pipeline. And this is sort of a, example. So first of all, we would want to standardize the acquisition of the data. And sometimes an intensity normalization is applied to the images. So this is like at the image level, it's a contrast harmonization, but it may not explicitly account for the batch. So you would want to maybe do this even within a given scanner, like scans from the same scanner. And then we have a category which I call statistical harmonization, you know, where we're also doing image level contrast harmonization, but it does explicitly account for the different scanners or protocols. And then we can also think about after feature extraction, like doing some kind of harmonization, which explicitly accounts for the batch. So ComBat is one example of that. And then finally, we can do robust downstream analysis, where we sort of pool the results in a robust way. So is it, for example, meta-analysis, mega-analysis, or hierarchical Bayesian regression? So I'm just going to give kind of a high-level overview and just give a few examples of each of these things. And just a point is that this area is, you know, a very active area of research right now. I've been getting, you know, Google Scholar alerts related to this topic, and just in the past 6 months, there's been quite a lot coming out. So that's really great to see. But you know, it's a rapidly changing field, and I don't know if, you know, we definitely haven't converged on a gold standard, I guess, and it might depend on the context of the study. So just to talk about standardized acquisition, for example, things to consider would be, you know, you want to have the same exclusion and inclusion criteria across study sites, obviously, I guess. And also, even if that's the case, you still want to also aim to balance the study sample characteristics across sites and scanners. So hopefully those distributions are similar. And standardizing imaging protocols and processing across sites and scanners is important. And as well as tracking any changes. Often, like, there might be scanner upgrades or software upgrades during the course of a study. So that should be tracked carefully if possible. I mean, and, you know, often, you know, establishing quality control measures across the different sites, such as using phantoms. And then there's traveling subject designs, where you have sort of a subsample of subjects which are scanned on multiple scanners across scanning protocols. You know, maybe if there's any changes in the protocols, you can scan these same individuals and assess the effects of those changes. And you know, these large scale studies such as ADNI and Human Connectome Project have put a lot of work into creating standardized protocols and quality control methods, and that's great work they've done. And I think it's a good point to consider, like for new studies, to consider adopting the same methods and protocols as some of these large scale studies whenever possible, so as to like facilitate, you know, the aggregation of those data sets later down the line. And there's just some selected references of, you know, discussing how to standardize acquisition. Right, and then this point I just want to make is that study design is key. And you know, you can do these harmonization methods, but they can't always fix everything. You can't do complete magic. I mean, we're trying to, but and there's of course the famous quote by Ronald Fisher about, you know, you can't revive a study from the dead. Really, you could maybe do a postmortem analysis, but sometimes you just can't fix it if the data was not collected in the best way. Okay, so intensity normalization, I am thinking of it here as more of an inter-image approach than an inter-batch approach. And I guess the original motivation for this is that MRI uses arbitrary units, and but we still might want to define biomarkers that involve image intensity metrics. So we want some way to standardize these intensities across images. And there's been many methods proposed to do intensity normalization, starting, you know, maybe in the late 90s with the histogram matching. So those are good, but it's found that the intensity normalization alone can be inadequate when we have a lot of across site differences. So there's other methods to supplement maybe these normalization.
Hi, Joanne. Would you take a question, please?
Oh, sure.
So how does MRI use arbitrary units?
Just like the values that come out in the image, as I understand it, they're not really scaled to a absolute scale. And like, I think this might be some of the idea behind scanner drift. Like even within a scanner, if you scan the same person, you know, you're going to get different intensity values for their brain scan, just because I don't know what the physics is of it, but there's no way to, you know, create an absolute scale coming out. Maybe somebody else knows more about that, but.
Yeah, there are no units on most of the scanners.
Right, so
like
they're
T1 weighted image, there is no units, it's just some number. And you can arbitrarily scale that number using electronics and amplifiers and stuff like that.
Okay.
right. So yeah, so these methods are trying to create a absolute standard somehow by using, usually you have to segment, I think, the brain and then into tissue types and all that, and match the distributions across scans or something. So here's, yeah, an example of a comparison of different normalization approaches. And you can see the different tissue types at the bottom, CSF, gray matter and white matter. And yeah, each line is a different scan, different image after the, yeah, okay. So now I'll mention some statistical harmonization, image level contrast harmonization that explicitly accounts for batch. So deep harmony is a deep learning approach. And it uses like this unit architecture. And it harmonizes, here's an example of two protocols. And on the left is the acquired, and on the right is the deep harmony harmonized. And here, And then after they did this harmonization, they did some segmentation, I guess, to get these volumes. And you can see that after deep harmony was applied on the right, the trajectories look much more harmonized. I mean, it's, yeah, so so some advantages to this type of approach would be that, you know, you're harmonizing at the whole image level, so you can do various downstream analyses. And they did see improved consistency of the volume quantification across protocols. This approach does work for longitudinal data and also kind of denoises the images. But you have to have an overlap cohort to train the model. So a subset of subjects that are scanned on both protocols are necessary. But I think they're working on creating a version that doesn't require that. And then just one more I'll mention is this mica, which is, you know, not deep learning, but maybe more of a statistical approach. It's a multisite image harmonization by cumulative distribution function alignment. It does a non-linear transformation of the image intensity values to remove scanner effects, and it aligns the cumulative distribution functions, and it does explicitly account for the batch. And it I guess one maybe downside is that it just removes the average scanner variability, which is an imperfect solution, but it is simple and easy to apply. And here's sort of an example of a single subject that was scanned, you know, multiple times on multiple sites. And, you know, even within the same subject at the same site, there's differences in the raw voxel intensities distributions. But then after, here they applied white stripe first, which is another normalization method, and then applied mica, and they get something that looks, you know, very similar across sites and scans. Okay, and then what about harmonization after feature extraction? So again, I'm just touching on some highlights. So here's one that actually it's maybe during the feature extraction process is when the harmonization is occurring. So it's a harmonized atlases approach. And this was done using the Baltimore Longitudinal Study of Aging data set, which had I think it had a scanner upgrade like during the study. And they wanted to be able to combine the data across these scanners. So, and here they're doing a brain parcellation context, so looking at ROI volumes. And so this approach creates a site-specific atlas, or I guess a scanner-specific atlas, and uses a multi-atlas label fusion framework to arrive at the volumes. And they found improved longitudinal consistency of segmentations. And here's some of their results. So on the left, we see the solid lines are the harmonized, and they put age on the x-axis here, and the total brain volume on the y-axis. And you can see that after harmonization, the two solid lines are closer together. You would expect them to be, you know, really the same ideally. And then the unharmonized is the dotted line. And yeah, on the right, you see the unharmonized and harmonized trajectories. Okay, and then I will discuss ComBat, I guess, in more detail, because that's something I've been working on. So ComBat stands for combating batch effects when combining batches. It was originally developed for genomics data in this biostat statistics paper. And it harmonizes means and variances of residuals across batches. So it's a type of location scale harmonization. And one thing that is necessary to do is to include covariance to specify the variance you want to preserve. Like, for example, age, sex, and diagnosis. And this is particularly important when covariate distribution differs across the batches. And the ComBat is, I guess it's distinct from just like adding a scanner covariate into your model or something, because it uses this empirical Bayes approach, which borrows information across features. So there's the assumption that scanner effects in one part of the brain will be similar to the scanner effects in the other parts of the brain. And so also, the effect is that this is robust to outliers in small batch samples, which was also, an issue for genomics data. So here's an example of how mean and variance are shrunk within a given batch across features. So the picture on the left shows the way the harmonized data ends up being shrunk towards the mean, I guess. And on the right side here, we can see the original data. If we plot, gene 1 and gene 2 against each other, we have an outlier in the upper right. If you just do a least squares approach on this data set, you get a distortion of that distribution, whereas ComBat adjusted was able to preserve the, that outlier actually. And the basic steps in ComBat are, first of all, we're assuming that the side effects exist in the mean and variance of the residuals of, and we're always going to have multivariate observations usually when we're doing ComBat. So we have, our Y here would be like something like cortical thickness in a given region, brain region for a given batch subject. And feature would be the region. And this X beta are the covariates of interest, like age, sex, diagnosis. And you want to be sure to, put whatever your hypothesis test is going to be, like some kind of diagnosis will be in that term there. And then we have this gamma, which is the additive batch effect, and this delta, which is the multiplicative batch effect, which is multiplying by the error term. And we impose priors on these batch parameters. So we assume that this gamma, the additive batch effect, is coming from a common normal distribution across features for batch I. And for the multiplicative batch effects, we can assume that it's inverse gamma distributed. There's also non-parametric version of ComBat where you don't necessarily need to have these parametric distributions. And then this is sort of the harmonized data at the end. So you just subtract off the additive effect and divide by the multiplicative effect, and then you're adding back the biological effects of interest. So there's been a lot of work sort of adapting ComBat to neuroimaging. So like J.P. Fortin used to be a postdoc in Pensive, and he did some of the first work translating this to neuroimaging data. I think he had a background in genomics, so he wanted to do this. And people have been talking, I think, about some problems with the code implementation. There was, I think Nick Cullen had a repository with a ComBat implementation that there were problems with. So JP was, I guess, very dismayed about that. So naturally, but I think this one at his GitHub is working correctly. And so Mei-Chen, you did some studies with fMRI using ComBat. And then recently, I worked on this longitudinal ComBat extension with my postdoc advisors. And Ray Pomponio is, people have already mentioned some of this work, extended it to the generalized additive model. And we're working on generalized additive mixed model implementation, and ComBat also harmonizes feature covariance. So not just the mean and variance of the individual features, but the covariance between the features, which we can still see scanner effects after applying ComBat in the covariance. So if you're doing multivariate pattern analysis techniques, you might want to consider adjusting the covariance. So he has a preprint.
Joanne, I have a question on the last slide. So,
Sure.
Sorry, the last slide.
Which one?
So in step 1, how do you find the residuals?
Oh, residuals. Well, I kind of left out a step, I guess. So normally you put scanner in as a fixed effect first. And let's see, I'm trying to think. Well, you want to basically first control for any variance due to the biological effects of interest, so like age, sex, diagnosis, that kind of thing.
I'm sorry
Right.
for interrupting,
So,
guys, but I think you can discuss this later in the gather time if
Okay.
you don't mind, because
Yeah.
we are kind of tight in terms of time. So
Okay. Sure. Sorry if I'm going too slow. I'll just skip over some of these. These are just unharmonized and after harmonization in longitudinal. Here, this is ComBat GAM example, where we have nonlinear age effects. They harmonized a very large lifespan sample. And this is a very cool, shiny app you can go and look at. And actually, Andre Marconstock was saying you can't harmonize new scanners, but actually Ray did find a way to able people to upload data and then harmonize them to the lifespan sample. So that could be done. And then this ComBat, it assumes covariance scanner effect is contained within the variance of the principal component scores. So he found that even if you do ComBat, you can still detect the scanner manufacturer, but after ComBat, it's less able to detect that, and also better able to detect diagnosis and sex. So there's many advantages, which I've mentioned. Disadvantages that you do need to carefully specify the model and account for confounders and control for imbalances, and explicitly preserve biological variability of interest. But one thing you can do is also just train the ComBat model using only the control sample. So if you do have like a bunch of psychiatric patients and you want to preserve variability that's unknown, I would probably maybe do this control approach and then apply the harmonization to the other. The harmonization model should ideally match your final model exactly. And I actually, this one isn't the case really. This last one, as I showed with Ray's example. So again, I just want to stress this again, that study design is important. And then for robust downstream analyses, meta-analysis and mega-analysis. So meta-analysis would be when you first estimate effect sizes for each study separately, and then you enter these into higher order models to aggregate the estimates. And then mega-analysis would be pooling data across studies and then using, putting it all into the same model. And here's an example where they tried a few of these techniques, meta-analysis and mega-analysis, and then they did a ComBat. It was with the Enigma data, and they showed, in this case, ComBat mega was greater, more powerful, I guess. Hierarchical Bayesian regression, I kind of put into this robust downstream category. They, in this paper, compared mega-analysis. I mean, I kind of putting these categories on here. And to me, this approach is similar to like a meta-analysis approach, this HBR. I don't know if the authors would agree, but it seems to me similar. And we've already seen this earlier talk. Just to finish, advantages of this approach, as was mentioned, are it's well suited to normative modeling. It can preserve biological variability, like you don't need to pre-specify that. You can add in new batches without retraining the model. But I guess some questions I would have, or things that I would think would be interesting to look at would be what happens if there is substantial confounding of important biological factors across the batches. Do you get the same normative model coming out if, for example, like sex is distributed in a balanced fashion or imbalanced fashion? How would that change your normative model that comes out, even if it's, let's say, the same exact subjects overall? And I wonder if there is a way to leverage within batch information across features in the way that ComBat does. And how applicable would this be outside of normative modeling? I don't know, but it seems a good approach for normative modeling, and I look forward to looking at the implementation of this. I think there is code available. Okay, and then that's basically it. I have a lot of references. If anyone wants to, I would be happy to share the slides. Thanks again to the organizers. Thanks to my postdoc mentors, Kristen Lin and Taki Shinohara, and everyone in the Pensive group, Hao Cheng Xiao for some slide content. And I've been having some great collaborations with Nick Tustison, who works on the ants, which he of course thinks is much better than FreeSurfer. I don't know. And Phil Cook. And those are my contact information. Thanks so much.
Thanks, Joanne. Thanks for this wonderful talk about the different methods to harmonize the data. It was wonderful, really. So since we are a bit beyond time, I think we can pass all the questions to Gather Town, or maybe they can just reach you directly, right?
Yeah, absolutely. Yes,
okay.
Let's talk later.
Okay, nice. Again, thank you very much. So let's pass to the next speaker, who is Yogesh Rathi. I don't know if I pronounce it well, I hope so. Yogesh is an associate professor at Harvard Medical School. I mean, super nice to have here Yogesh because he's an expert on diffusion data. He has also recently published a preprint on, the ways or the benefits of improving power in neuroimaging studies, harmonizing diffusion data. So Yogesh is going to talk to us a little bit about, the different harmonization methods to diffusion data. So it's all yours, Yogesh.
Thank you, Javier, and thank you for the organizers for inviting me for this talk. I tried to be very general in my talk as well, but focused much more on the diffusion. So everybody can see my slides? Yeah, all right. Okay. As I said, I'll go a little bit on the specifics, and I think that was a great talk by Joanne. She gave a lot of the background on why we have issues, but nevertheless, I have it in my slides. I'll go over some of the issues, but skim through them a little bit faster. And very briefly, why harmonization? And I make some distinction between harmonization and data pooling or statistical pooling. And today I will have only time to really very briefly talk about retrospective and prospective harmonization, but I'll focus a little bit more on the challenges and the open questions, and I hope that will stimulate a lot of discussions. So reproducibility is a key. We want reproducibility. We want that same subject scan on the same scanner to have exactly the same type of data. However, this is an example here. You can see this is the same subject on the same scanner, scanned twice. On that picture, you see that, and again, as I said, I'm focusing purely on diffusion MRI data here. So the variability is really low. I mean, there is some variability within the scanner about fractional anisotropy, which is one of the key measures of diffusion. So about 2 to 4% variability in the white matter, maybe, and then 10% in the gray matter. And on this plot, this is again for whole brain for generalized fractional anisotropy. You have 3 subjects scanned on 2 different scanners. There is some difference between the scanners, but within scanners, they're pretty good. So why is there even that 2-5% variability? So because magnetic field inhomogeneity, differences in shimming that can happen, biological variability exists. There can be pulsation effects, also impact of scan time of day. So there is a work by Thomas where if you scan the same subject, let's say in the morning and then in the evening, you should expect they were able to see some biological effects. Because of the circadian rhythm, you have different effects there. And then there is gradient performance and including scanner drifts and so on. So that also produces variability. Between scanners, here is an example of the same subject scanned on 2 different scanners, Siemens versus GE, and you can see the percentage change in FA. You see at least a 10% difference in FA in the white matter, 20% in the gray matter, and more in the CSF. While we don't care so much about the CSF, but significant effect, 10%, 20% at least. And this is the same subject, same scanner. So why do we care? I mean, okay, 10, 20% difference, so what's the big deal? Here is, I'm just taking some 2 representative examples here. 2 groups of subjects here on the left, you see the difference is really 5%. And this is more often the case in psychiatric disorders. On the right is a very high profile paper between swimmers and soccer players. And what you see is that, you know, although at the first sight, you would see that the differences are really large here. Actually speaking, they're not. They are 2 groups that are very tightly bound, but they're separate. But the difference is 14% only there between these values on an average. So what we're saying is that if the group differences we are looking for is 10 to 15% at the most, then, and if your group variability is 10 to 15%, or sorry, within scanner, between scanner difference variability is 10 to 15%, there's a good chance that if you were to scan these set of subjects on different scanners, there is no way you find any of these things. So obviously we need to remove these. And this is another example from our recent work. You have 3 different groups that were scanned on 3 different places. And if you were to not do any harmonization, you would see this yellow colored dotted line here as your analysis of how a particular measure called FA changes over the whole brain across ages. It's a cross-sectional design, but you can see it would be a completely wrong conclusion compared to what you should be getting, which is this purple line, which is what we know is the case. So quickly going over inter-scanner differences. Although I might be repeating some of these, I'll go a little bit faster on these. But just to clarify a few things, differences in magnetic field inhomogeneities, there is differences in number and sensitivity of the receiver coils. Then there is vendor-specific differences in MRI reconstruction algorithms. These changes are not, okay, one single linear change or bias across the brain. It's not that case. You can see here, these are region dependent. These are specific different types of tissue, very specific for different types of tissue. And this is very much so for the diffusion MRI data. It exists in all frequency ranges in the diffusion MRI signal. So unlike T1 weighted and T2 weighted images where you have only a single scalar, here we have, you know, the data is 4-dimensional. So a lot of different changes exist in all frequency ranges. And these are highly nonlinear. So one cannot just take a physical phantom and say, oh, I'm going to scan the phantoms everywhere. And I'm going to just take that and apply it to my apply it to or use it for harmonization. You can't do it just because it's too simplistic. You can't, there's no clear way how you will take differences in phantom. Let's say you find differences in diffusivity. How do you translate it to differences in this brain, for example, this image here? It's not possible. So I think here you're sold on why we need harmonization. So I'm just going to go through it, brush through it. But our goal is to try to see if we can reduce our inter-scanner variability to intra-scanner variability. And some specific thing is to ensure that fiber orientations and everything else stays the same when you do this harmonization. This is important for diffusion MRI data in particular. And so now I'll go a little bit slightly just to provide details about what people have done so far in diffusion MRI or elsewhere also.
Yeah, Yogesh, you don't need to hurry. Take your time, okay? Take your time.
Sure. Okay. So thank you. So there is meta and mega analysis. I think these were very often used very early on. They became, naturally, they are very good ways to get around the problem of scanner differences. So what you end up doing is you calculate a z-score at each site, and you compare the z-scores rather than computing the measure of interest. So for example, if it's fractional anisotropy or if it's cortical thickness, you calculate the z-scores within your cohort of that site, and you compare the z-scores directly. But this will be a have some potential confounds here, because if your sample size is small at one of the sites, then your variance, because z-score has this in the denominator is the variance of the sample, and if your variance is not really captured properly, then all bets are off as to what you find there. Again, also it assumes that everything is linear between the sites and between the regions of interest being analyzed. But I think it is a great method if and more practical method if sites cannot share the raw data or derived data. They only want to share the z-scores. Well, this is how I think the Enigma, I mean, they also share the FA values, but they don't necessarily have to share the raw data, which is a bigger issue with privacy concerns and stuff like that. So in that case, this works out pretty well. Then there's these methods of adding statistical covariates. Again, nicer methods, linear covariates at each voxel, which is not the case with these meta and mega analysis because they have to be done region-wise. But in statistical covariates, you can at each voxel, you can add a statistical covariate. It definitely captures the regional and tissue-related inter-scanner differences. But again, the assumption is that all of these effects are linear, which may not be true. And I think we show that in several of our papers that it is not true, but especially in the case of diffusion. Maybe true for certain modalities, especially, for example, maybe T1, but certainly for diffusion, it's a problem. Then there is method by Killian Paul. What they did was to say, well, we're going to have 3 traveling subjects. And we have these three traveling subjects at each of the sites. So we're going to just use these 3 traveling subjects in a region of interest. I'm going to estimate my measure of interest, let's say FA or any other measure, and I get a correction factor. So basically a linear correction factor. And I'm just going to apply that correction factor to everybody from that site. And that's how I go for it. This method will work well if all the subjects were obviously scanned with the same type of head positioning, etc., which is never the case. I mean, you typically have people place their heads in very different setups and formats and places, and obviously it also has issues with one of the recent UK Biobank papers that showed that where the subject's head is in the isocenter of the scanner also matters. So these things will be a big potential confound. And then you have large ROIs like corticospinal tract. In the case of diffusion MRI, that is the big advantage of diffusion MRI is you can trace these fiber bundles going from say the brainstem all the way to the cortical motor regions. And the scanner differences, as I mentioned and showed in the picture earlier, are not the same across the brain. So then how do you use these type of correction factors would be a question. And I think ComBat, Joanne really gave a very nice overview of ComBat. So luckily I didn't put any of the equations. I removed all the equations from my talk since it was going to be brief. But I just want to say we also use this original version of ComBat from 14. It has, it is much better than all the other ones I mentioned earlier because it has two different factors, additive and multiplicative factor, to account for the site effects. Although it assumes some prior distribution and Gaussian inverse gamma on the site effect parameters, which may or may not be true. Especially in diffusion, things are a little more complicated, so they may not be true at higher order frequencies. And so this, and then one of the key effects that we also see is that, and I think these are being addressed in the latest versions of ComBat. So I think there are at least 5-7 different versions, newer versions of ComBat now coming in that address some of these issues. And one of them is what if the your regression coefficients or the some of the different dependent variables are not linear. For example, age. As I said, fraction and anisotropy varies very non-linearly with age. What happens then? So one may, we cannot regress that out by linear methods. So you can add non-linear methods. But many times that entire nonlinearity is again dependent on your sample in that in what you have. So sometimes that is a little bit tricky. And then again, some of the other things that people have talked about is, of course, it needs a very accurate registration. So what if there is large atrophy or larger ventricles from older subjects? Because registration is not perfect. And what happens there? So there will be some of those, I think, questions that are being, I guess, every few months, some of these questions are getting addressed by newer and newer methods. I haven't discussed all of them, but I want to just let people know what are the good things about each method and what are some of the limitations. So our method that we worked on has been on harmonization of the raw data signal itself. In this case, what we do is you either have traveling subjects or match controls from each site, and we learn what is the scanner effect from the raw data itself, from the raw diffusion MRI data. It is at voxel level, so regional and tissue specific effects are taken care of. It also is nonlinear in the signal domain. So for all frequencies, things are taken care of. But it has one limitation, or at least one limitation, I would say. And that is that you need a sufficient number of match controls at each site or sufficient number of traveling subjects from each site. Otherwise, it will not work well. So you need these matching controls, typically, which are required in most cases. Not all cases, but most cases you acquire a set of controls. And so the field of harmonization, even diffusion MRI, is also growing fast. You can see this is a review paper. And then there are so many new methods that are being proposed almost, as I say, every few months. And each of these methods addresses certain different ways of addressing the harmonization issue. And what I would like to draw some attention is some slight differences between the class of methods. So what I call data pooling are really statistical methods. They account for scanner differences for each different diffusion measure. You need to run them separately, and they do it in a slightly different manner. And they're done at the very last or at the very end of the pipeline, in the sense that once you do this harmonization, then you do the final analysis of what the statistics need to be. But there has to be a significant amount of pre-processing done before you get to that point, especially in diffusion MRI, where you need to do eddy current correction, EPI distortion correction, but that is pretty standard. But then you still do denoising and or model fitting and or retrography and so on. And after that, you need to do harmonization. Whereas in the methods that we have been looking at, they do it at a different stage, and they're directly applied to the raw data after pre-processing, which means motion correction and EPI distortion correction. There has been some recent interest in doing it this way, partly because I'll list some of the reasons here. So I think some of these things have not been explored completely by the community, and this is still open challenges. We don't know how these unknown scanner-related nonlinearities propagate through the processing pipeline. We have started to look into some of these things, and I have some slides, but I can go deeper if anybody has questions. But what if somebody changes, or what happens if two different processing pipelines are used? Would you get the same results from ComBat, for example? That's a question, because you're making so many design choices. I think, as you've seen many presentations, you have to make design choices or processing pipeline choices, and depending on what choice you make, things can be different. Modeling assumptions. So how do you know that if you use a single tensor versus multi-compartment model versus any other model, that the scanner related noise is not affecting your fitting, and as a result, affecting the ability of ComBat to harmonize or any other method to harmonize? And so, as I said, noise characteristics also play a role in these type of things. So here is one example I'm giving you. Here it's the same exact data set. There are 5 different sites. For each site, we are running the processing the data using 3 different software. Processing in the sense only the DTI fit. All other steps were constant. Only one step was changed, and this is what you see already. At least out of the 5 sites, 4 sites are fine, but one side you have a significant difference between just the data fitting, and that is because of noise. You can see that in FSL there is different compared to MR tricks compared to slicer. So the question is, how would, let's say, if somebody does it this way, let's say one site processes the data with FSL, another one with MR tricks, third one with slicer, even though they're doing exactly the same thing, they think they're doing that with a DTI fit. They're using the same ordinary least squares fit, and this is at the whole brain level. How would anything work? This is our comparison with data pooling. So one of the key things that I would say is that no harmonization work should change effect sizes. They should be same after harmonization, right? If you have a particular effect size before harmonization, that should be the same at after harmonization. And you can see that here on site 2, before and after harmonization, the effect sizes are pretty much the same. I mean, this is great. And this is between healthy controls and schizophrenia patients. But on the right side, in site 3, this is again using the same exact processing pipeline, even including using the same FA value, diffusion tensor estimation software. You can start seeing differences here. I mean, look at the right SLF. You have after harmonization. I mean, you can see differences in the harmonization efficacy.
Sorry to interrupt. Could I ask a quick question on this?
Sure.
I saw this poster at OHBM. I was interested in those. Do you think those effects are statistically significant, those ones that are flipping? And also, is scanner or site used as a covariate in the before analysis?
No, in the before analysis, we did not. Sorry. In the before analysis, it's the exact same site. So there is no differences in site. It's one single site. What we did was to say, okay, let's take this site, this data, and harmonize it with another site. And after harmonization, we just took the same exact data. So before harmonization, we knew what the effect size would be. And that should be the case after harmonization also, no matter where you harmonize it to. So that was the first thing. Second thing, about effect sizes, whether these are significant or not, we did not test for the statistical significance. But regardless, I think I can show you some other places where I would almost expect that there will be significant differences between, because the effect sizes were just so large. But having said that, I say that we did not do a specific test to say that they were statistically different. Okay. Yet we thought that it shouldn't happen, even though effect sizes should not flip, is what our thought was.
Okay, thanks. I'll talk to you more later, maybe, about it. Thanks.
Absolutely, absolutely.
Yeah, quick question, Yogesh.
Yes.
Why? So are you implying the effect sizes for before are some sort of ground truth here? And because they're not harmonized, how can we trust them? Like we know harmonization was necessary to estimate the effect sizes, right?
So yeah, so let me clarify. So let's say you take a large, let's say what we did, I'll give you other examples also. Let's say what we did was we took one site, let's say the Philadelphia Neurodevelopmental Cohort, PNC data. It has, I don't know, 800 subjects. We harmonized, we took males and females, for example, and calculated the effect sizes. It is the data calculated, the data acquired on the same site. So there is no site effect there. Okay, they did not do any upgrades. They did not do anything else, right? This is a simple study. Let's say you would do this study by saying, I want to calculate the difference between males and females for a particular variable. And I calculate, okay, this is, maybe whatever it is, FA is different in the SLF. You calculate the site effect. Now you store that. Now I have another site on which a similar age group was acquired, let's say Brigham and Women's Hospital. I want to now combine these two data sets and I run ComBat on that. So after running ComBat, the site effect would be removed between BWH and PNC. Now what we do is after that site effect is removed, we take away the BWH data again and only keep the PNC and again compute this effect sizes. So now there is, after harmonization, there is some change in the data. Now, if we do and go ahead and calculate the same male-female group differences, they should be the same because the harmonization shouldn't touch them. The biological effects should be the same, unless something happened and the effects of not only scanner but biological variability were also affected. And that is what we are trying to characterize. So I think this is a very important thing because when you're saying I want to use a particular method, one of the key things I want to test is it going to preserve all my biological variability or group differences, or is it going to mess up with them? And I think, yes.
Yeah, that's fantastic. In fact, actually, I would love to use any open source data towards testing of the library I'm trying to build. Because one of the tests I want to put all the methods through is to see if they introduce a bias that did not exist in the original data.
Absolutely.
That should be a test for all the harmonization methods. And we should try to develop an open data set so people can do regression tests on this,
Absolutely, absolutely. So I think there are going to be a ton of data sets that are going to be available. I think the NIMH data archive is one of the places. The lifespan and the human connectome subjects are another one, and PNC data is also publicly available in the sense that I think you can apply for it and you can get access to it. It's a ton of data sets I think I can point you to more when we look into it.
Sorry, Yogesh. So maybe what you mean by preserving the effect sizes is the sign right here, because
Not just the sign, the magnitude too.
the magnitude.
So,
but there are papers saying that when you apply the these harmonization methods, they are supposed good to be good to to amplify the effect sizes, right?
I would not believe that.
Okay.
That I would not want any amplification or any change. When I say that, so if I, as I said, let's say PNC data, I have the original data. If I were to not do any harmonization, my effect sizes are what my effect sizes are. I would have to report them in a paper, and that is what it is. So after harmonization, why should there be an increase in effect sizes? Wouldn't that be amplifying something that doesn't exist in the original data?
Or maybe you are getting rid of unwanted variability, right? And
I don't know if it is unwanted. How do you know?
Yeah, no, you don't know for sure. Yeah.
Yeah. So this is where I think that we have to be very careful. I think there has to be more emphasis on doing a little bit of these type of analysis for every paper that comes out on harmonization, to say, are you going to preserve your effect sizes? Are you going to change them in some way? Even if you change them, what is the quantity? What is the magnitude? For example, I'm very happy with site 2. There are minor changes, I don't care so much. But if there is major changes, I do care. All right. So I think this is just a very brief slides that I earlier talked about physical phantoms. I think there is a study by Timmermans, which is a very nice study. They use a phantom to show that almost all scanner variables produce variability. Coil, scanner drift, bandwidth, software, vendor, etc. You name it. So, and I have to say that what we get is the data final product out. When we get the data, it is not a linear combination of these things. It is a very non-linear combination of these things. So I would be very surprised that we will be able to very robustly measure the scanner effects just by linear models. And so I think even if this paper used a linear mixed effect model to quantify these site differences, it is not clear how you translate them to human brain. So I'm not sure, but maybe most of you know there was a community challenge. There have been 2 community challenges on diffusion MRI harmonization. It's called Mushak Challenges. It was held with mica, which is a conference, and at a workshop called Computational Diffusion MRI. What they did was to say, they gave us 10, they had a total of 15 traveling subjects on 2 different sites, or 3 actually speaking, but 2 is what we consider here. 10 of them were given out for us to figure out a learning or mapping or whatever we want to do with it, and 5 were kept for testing. And they were not given to the people. 21 of different algorithms participated in the challenge, and one of the methods that our result was this is just a sampling of the difference between the scanner after harmonization, and this is the same subject scanner, multiple scanners. So you, as I said, you don't want any kind of effect staying away. So what we see is that our result was pretty much very, very reduced the error. This is the absolute predicted error of FA, and this is mean kurtosis. As you can see, the difference between on those 5 testing subjects between those scanners was dramatically reduced. And I think these results are available in these recent papers in Neuroimaging. So what are our current directions? We are currently harmonizing the ABCD data. It is a retrospective harmonization, more or less all of these cases, but these are 10,000 plus subjects that were just released by the ABCD study. They are from 40 sites. We are going to harmonize them as part of our R01 grant. and deposit them back to the NDA. These are only diffusion MRI data. And then we'll do the same thing with the human connectome and the human connectome disease subjects. And there is 10,000 data sets from the Healthy Brain Network, which is supposedly, if it was not COVID, they would have had some traveling subjects also, but they're not doing it anymore because of logistical reasons. And our goal is to perform harmonization on all of these subjects and deposit them back to the NDA. We will also do multiple analysis on them. Like we'll extract out specific fiber bundles in the brain and process them and release them as part of different networks of the brain, but individual microstructure measures and everything will be also provided. So I think some of the learnings we have or thoughts we have is that it's better to harmonize earlier the signal itself rather than at the very later stage, because we don't know what kind of effects might affect our analysis. And in the case of prospective studies, use of traveling subjects will be really good. This type of analysis also allows tractography. I think this is one very key important point I want to emphasize as much as I can. That let's say you were to run tractography on two different data sets, which are from two different sites. Because the noise levels are different, because the bias are all different, the tractography itself is going to be different. And then you try to harmonize may or may not be really a valid harmonization approach, because you might have already missed out on a part of the connections, or that your tractography itself is biased, and then that bias cannot be captured by post statistical pooling methods. So in this case, harmonizing at the signal level will be much better, because then at least the signal is consistent. There are some open challenges here, which I'm also going to talk about. We want to ensure that we are not introducing any new information in the data when we do the harmonization. For example, you're not adding new peaks or changing the original orientation of the fiber. That is extremely important. We have tested it in our setup, but any method that does this type of analysis should make sure that these things are tested for. Validation should be done on not just one metric, but a whole bunch of model independent metrics. That is very important because, again, as I said, models are models, and it is better to have model independent metrics to test your algorithm, like generalized fractional anisotropy, orientations, etc. Although FA and mean diffusivity are model-based ones, they are some of the basic ones I would test, but also other model independent ones. What effect does site-specific noise distribution have on your harmonization technique is something that needs to be characterized. And there are also, similarly, as I said, there are open questions for data pooling too. And this is very specific to diffusion MRI. So let's say you have two data sets with very different B values. And we know that the relation between B value and fraction and anisotropy, or MD, is nonlinear. Can any statistical adjustment really work in these scenarios, linear ones? Because your partial volumic effects will be very nonlinear, and also just that FA would be a very nonlinear function of the B value. And if you're just directly using FA, how do you say that it is you're only capturing site effects? Spatial resolution, again, the same story goes. There could be other types of biases. Then what if what kind of bias exists if some of the sites do not capture the entire normative biological variability? So you have 5 sites and 2 of the sites do not have a larger data set. How does that affect meta-analysis and mega-analysis? And what about nonlinear variability or nonlinear bias due to other demographic or clinical variables like IQ or age or cognitive abilities. So those are things that we need to consider about. And again, what if your assumptions are not valid? I think this question is valid not just for data pooling, but for other harmonization techniques as well. I will stop here, and I'm thankful to Suhaila, who's my postdoc, started with me on this harmonization journey, and she's really the one who drove and tested and developed all these algorithms, and Tashrif, who has really converted all of that science into something more usable in terms of this harmonization software, which is available on GitHub. And he is always very active in helping you answer any of the questions. So with that, I'll stop. And if you have other questions, I can delve into other slides. Thank you.
Okay, thank you, Yogesh. This was a fantastic talk. So yes, I think we can pass all the questions to Gather Town, direct to you later. So before passing to the next speaker, I would like to know if if there is some hurry in the audience, especially to the next speaker. We are going a bit slower than we expected, so we would like to know if it is okay with all of you going like this, or if you have some kind of hardness, something. Okay, so if not, I think we can go like this.
A quick question to everybody, actually. So from what I can tell, everybody is familiar with Git and GitHub, right? So I'm thinking perhaps we can skip the tutorial to save some time, also because our speaker ran into some trouble with charging and whatnot. So raise your hand if you have no familiarity with GitHub and you would like a tutorial. Or maybe should I ask poll, or maybe type something. Okay, because I don't see any raised hands or chat or whatever, we'll skip that. Thank you. So the reason we're going slow is to give you a chance for discussions and debates, try learn when the experts are here. I learned a lot actually, and I would love to talk to Yogesh and his lab more. So over to you, Javi.
Yeah, no, I mean, indeed, these were two fantastic talks. I mean, it's normal that, that we are taking a bit more slowly because they brought a lot of important topics. Okay, so let's pass to the next speaker. It's a pleasure to meet, to introduce Richard Dinga, who is a postdoc at the Donders Institute in Netherlands. He works with Andre Marquand, our, guest lecturer this morning. Richard is an expert on the application of machine learning to clinical neuroimaging data. He's also an expert on the confounding neuroimaging data. In particular, he has recently published a preprint about controlling for confounded effects at the machine learning prediction levels, which I read and really enjoyed, and I encourage you all to read. So today he's going to to tell us about a little bit more about different deconfounding methods when doing predictive modeling. Okay, so Richard, the floor is yours.
Thank you. I will start right after I find out how I share this. Okay, does it work? All right. So yes, I will talk about confounds in predictive modeling. Thank you for your introduction. That means I can skip all of this, except that this is how I used to look like before my PhD. In this presentation, first of all, I'm afraid that looking at the caliber of speakers I saw today, it might be too easy for you, but we'll see. But I will be talking about confounds and their issue for machine learning. And there are three messages. One is that, one will be that even though some people say that confounds are not an issue for machine learning, it very well is. Another point is that the harmonization technique and input adjustment technique probably won't work for the machine learning models, although I just find out about the ComBat method from the previous speaker, so this is something to worth looking into. And the last one, that kind of the usual statistical methods that are not like harmonization, they will work very well unless you do something better. So, but first, and what do we mean, or what I mean in terms of this presentation by confounds? So it's some kind of third variable that is related to both my input variable and my outcome. And the nature of this relationship is, for this presentation, is not important. So it can be side is related to age, but it doesn't mean that if you get scanned in one side, it would make you younger or anything else. It can just mean that there is a sampling bias, that some people just, the older people are scanned in one side and young people in the other side, which is something we observe very often in real data. And for imaging data, usually what we deal with is age, scan site, BMI, motion, things like that, which are often very related to clinical outcomes or some other biological outcomes of interest. As I mentioned previously, some people, here, Stroman, might say that for machine learning, we don't care about confounds at all because we just want to make good predictions. And if the confounds predicts my outcome, then it's good. It's all signal is signal. It doesn't matter that I'm predicting motion, if it then maybe helps to give people good treatment or something like that. It doesn't matter, even though it's motion or scan site or something like that. But there are a few problems with this approach or with this idea. One is that if we have a confounds in the data and we care on, and even if we care only about good predictions, but if we have confounds in the data, the results won't be reliable and then won't generalize. So imagine that I'm not really predicting the diagnosis, but I'm actually just predicting scan site. And in one scan site, I have more depressed people than in another scan site. Well, this model will then generalize only if then in real data sets or in real applications, this bias will continue, but it will probably not continue, right? Because now I have bias there because whatever reason researchers collected the data in one way or another, but this scans, but it doesn't mean that now everybody who will come to get scanned in Amsterdam will be depressed and everybody who will become scanned in Nijmegen will not be depressed. So I know these biases that I learned because I have confounds in my data will just then break my performance in like real applications. It also works for the generalizability of the results. And another problem is that the predictions might just be suboptimal. So even if I'm happy that I'm predicting, let's say, diagnosis only based on motion, and I'm fine with it, I don't want to make any biological claims about what I found out, maybe there is a better way to measure motion than putting people in a scanner and make a picture of their brain every 2.5 seconds. So if I'm fine with motion, then now I can look into my data, find out that I'm just predicting motion, and then go and spend my time making a better motion detector or something like that. And then the last point is that even if I don't care about anything, still good to know what my model is actually doing. Even if I don't want to make any actions about it, I still would like to know if it's really driven by motion, if it's really driven by BMI or some other variables. And of course, if it is driven by BMI, motion, age, then maybe I don't need to waste time on the scanner. Maybe I can just make a good model based on BMI, age, or other variables. Now, so in general, what can we do to deal with confounds? So it seems like you all understand this, but maybe somebody in the audience. So we have to get rid of one or both of these effects. So effects of my confounds on the outcome or the confounds on the input variables with one way or another. There are many ways we can do it. So the harmonization methods is usually trying to get rid of the effect of the confounds on the image, making some kind of elaborate, more or less complicated model of the covariates to the image. But for the machine learning models, this might not necessarily work. It will probably improve your model and makes it better, but in terms of, am I sure that I'm now not just predicting confounds, it can be guaranteed that this will work. So why not? Well, because the machine learning models are probably capable to learning much more complicated relationship from your data than you can regress out using the traditional methods. So, for example, regressing out confound variables. So let's say I have two sites called 0 and 1, and before adjustment, their distributions of one voxel are far apart. And if I adjust it, that means that I basically centered it by mean, and now they shouldn't be really. I shouldn't be able to learn the information about site from this adjusted variable. But maybe it looks something like this. So after adjustment, there is a still difference in variance, and something like nonlinear support vector machine can learn this difference in variance. Of course, I might, if I use ComBat also model the variance as well, so this wouldn't be a problem for ComBat. But maybe something like this will be a problem for ComBat. So now the variance is the same, but kind of the shape of a distribution is different. And the support vector machine can still learn it. It's not, of course, it's not realistic. I hope so, but but as an illustration that there are other things in the data than just mean and variance that can still be very easily learned by machine learning methods. That may be a linear machine learning method or also nonlinear. Another problem is that if I'm regressing out something from the data, for example, centering by mean, and now I'm using, for example, support vector machine, which is not as sensitive to outliers than the model that I used to adjust for confounds. Now I can still, again, learn information from the data that I try to remove. And the last thing is that maybe, again, there is nothing else in this data set about the confound groups that I can adjust in individual variables. But if I put the variables together, now there is a pattern of this. Now there is a pattern that I can learn. And if I'm just adjusting one variable at the same time, I cannot remove this at all. Seems like this is a situation where the ComBat that was mentioned previously might work, but there's still the point is that it's not guaranteed to work, and there is always more complicated relationship that can be in the data that I can model and remove it. So, before, so I showed you that one way to deal with the way to deal with the confounds is to remove one or both of these effects. But this is actually a simplified image, and it can be like this. So I don't have to remove the effect of covariates to input variables, which is hard because there is many input variables and it's an image, and there are many things that my covariates can have, many ways that the covariates can affect the image. But to learn the effect of covariates on the predictions themselves, or maybe on the outcome, is much more easier because it's just one variable. And also, since it's just one variable and not 100,000 voxels, then I can actually check if my assumptions are met and all these things. And so basically what I do is, or what I propose everybody should do, is that now any method that you would use to deal with confounds for biomarkers, you can basically use with some caveats. But now the difference is that the biomarker I'm trying to adjust is not the whole image, but the biomarkers, so-called, I'm trying to adjust is the predictions I already made using the machine learning model. And so one way to do is just your ANOVA or ANCOVA and linear model. So just add the confounds to my linear model, and this would, assuming the assumptions are met, this would just deal with the effects of confounds. And it doesn't matter how complicated the effects of confounds is on the input variable on the image. It's enough that I get rid of this arrow on this stage. So if I go back, if I cancel this one arrow for machine learning predictions, then it cancels this whole path. So that's enough. If I want to be sure that I corrected for confounds, it's enough to get rid of this effect. I don't have to model the whole complicated effect of the whole image. So I can do ANCOVA or linear model. And what I can do is actually kind of like a analysis of variance type of decomposition of what I'm predicting. So here I'm predicting IQ and UK Biobank data using the brain image, the traditional some brain image predicting IQ. And I'm using, and with the linear model, I evaluate how well this model works. And I see, and my confound in this case is the brain size. So is my fancy machine learning model predicting something that I can't already predict, only knowing how big someone's brain is? And here I can see that, yes, this is the case a little bit. The most what I can say is in the brain size, but a little bit is also independent of the brain size. And I can do this, for example, on education. And here I see that education by itself explains much more variance than my fancy machine learning model, but there is still independent portion of variance that I can find out, that I can predict in the IQ using my machine learning model. So this is good to know, even if you care only about prediction, it's still good to know something like this is happening in your data. And so this is what we proposed in my preprint, and then there are other approaches. One is that you just deconfound the outcome variable, so you don't residualize the image, you residualize the outcome, which works. It's very similar to this full linear approach. It has slightly less power, and you don't have this nice decomposition of variance, but it will work as well, assuming the assumptions are met, of course. But it will work as well unless you do something wrong with like training set test side and things like that. Another thing is like a resampling approach or like a matching approach. So I have case control matching or propensity score matching weighting. This will work as well. I can do it, but again, I'm not matching the images or anything. I'm matching the people based on their covariates. And I don't care about the effect of the image. I just care about the effect of covariates and the outcome. But the problem is that I'm throwing away data, and there are some other issues with that. But in principle, it should work as well. Another thing is the permutation test. I can run my permutation test and shuffle people only within site or within H-bins or whatever I consider to be a confounds, which will work as well. Of course, you have to run permutation tests, but it's also good. And but the problem is that again, you don't have this nice decomposition of how much better my model is compared to model only based on that's predicting only confounds or something like that. But it will give you correct p-values that are not affected by presence of confounds. And this is pretty much it. There are some other caveats of what can go wrong. For example, in cross-validation, it's not so easy, or should you fit it only to controls or controls and cases together? You should do it together, but it's all these other stuff are in my preprint, and I thank you for your attention. And that's it. Yeah.
Thank you, Richard. This was wonderful. I think we have time for a couple of questions, if someone has. Okay, so I have one question for you because I read your paper and we really enjoyed it, and we actually started to apply this kind of approach, for controlling for confounders at the machine learning predictions. Maybe the problem that the question would be, what happens if you want to report the phenotype, the weight maps, emerging from any kind of linear models, if you don't control, for covariates or for confounders there must be some kind, you may be reporting some kind of shared variance, that should reflect, at the phenotype level, So what could you do, in that case?
Yeah, so this, so if you don't do anything before you get to predictions, then of course the weights won't be unconfounded or anything like that. To be honest, I'm not a fan of sharing weight maps or trying to make inference based on weights anyway. So for me, it's not a problem. But I know that people do it, and then then I don't know what you should do in that case. Okay. Probably you have to try to deal with the confounds before you get to the stage where you are making predictions. Also, if anybody wants to check the preprint, I send the link to the chat. So, thank you.
Okay, so thanks again, Richard. This was wonderful. So now